All right, folks, here we are continuing our Chapter 2 lecture series, the biomolecule component. We've looked at the carbohydrates and we've looked at the lipids, and our job now is to investigate the proteins. Now, your proteins are super cool, and your proteins have lots and lots and lots of important functions in the body. And so let's investigate that a little bit further. Now, your proteins, just to kind of give you an idea of how significant they are, they're formed by the direction of the DNA molecule. You probably have heard of that before. DNA is your hereditary molecule. So when we talk about inheriting traits or characteristics from our family members, what we're inheriting is the direction for how to make these proteins. So our proteins are the result of our DNA sequence. Okay, so that's the significance here. Now, examples of functions for proteins include catalyzing your chemical reactions. And we've talked just a little bit about enzymes and biological catalysts before. When we talk about enzymes being important in catalyzing chemical reactions, what that means is if you don't have the enzyme, you don't get the chemical reaction. So the cell can determine what chemical reactions can occur based on what enzymes are available. We even mentioned communication when we talked about lipids, and we see that proteins are also involved in communication. They're involved because some of our hormones that are not lipids are proteins or protein derived. Also, your cell membrane channels and your receptors are often made from proteins. So when we talk about a hormone talking to a cell or tissue, it is talking to that cell because it's binding to a receptor. And so you have to have that exact type of receptor in order to receive that communication. You have proteins involved in the structure of your body at every level. So as you are looking at your tissues in the lab, you are seeing collagen fibers, that's a protein. You're seeing elastic fibers, that's a protein. You're seeing reticular fibers, those are proteins. So proteins are going to be important in the structure of the skeleton. Proteins are going to be important in the structure of your skeletal muscle. Protein will be important in many of your connective tissues. I mentioned that proteins are important in the structure of your muscle, but they're actually the molecule in the muscle that is providing that contraction or movement. And this is true not just in your skeletal muscle, but your cardiac and smooth muscle as well. And then we will see proteins involved in your immune response. And they have kind of two different jobs that they do here. They act as these little name tags hanging off of your cells that allows your immune cells to recognize, oh, that cell belongs, it's not foreign, I'm going to keep it safe, versus the recognition of a pathogen that needs to be destroyed. When we talk about having an immune response and developing antibodies, those antibodies are proteins. So here we can see there's a whole list of things that proteins are doing for our body. So they're very cool and very important. Now, as we talk about our proteins, we've already discussed their functions, but what we have next to talk about is the structure. Now, the structure of the protein is the most complicated of the 4 macromolecules that we're studying. And why are we going to spend so much time talking about the structure of the protein? Well, because the structure is going to determine its ability to function. So in this case, this is a good representation of form meeting function. If in any way you change the shape or structure of the protein, it does not function. Sometimes that means it doesn't function as well as it should. Other times that means it doesn't function, point blank, period. Now, how is this protein structure determined by the cell? Well, number one is the DNA sequence. So we already talked about the DNA is that hereditary molecule. It's the information you're getting from your parents. And basically what the DNA molecule is telling your cells to do is how to make those proteins, the correct structure to those proteins so that they work correctly. But then we will also see there's a layer to the structure that is determined by the environment in the cell. So temperature affects protein structure, and things that are dissolved in the fluid of the body, electrolytes, that would be your ions, can influence protein structure, and pH can influence your protein structure. Remember way back we talked about homeostasis, which was the regulation of the internal environment of your body? Why is that so important? This is why that's so important. If you don't have your protein shaped correctly, so if you in some way alter the structure of your protein, you're altering its function. And it's either not going to work as well or not at all. Point blank, period. And if it's a super extreme change in the environment, that might be a permanent loss of that protein. As we are investigating our protein structure, we have 4 levels, and we're going to talk about it as primary, secondary, tertiary, and quaternary structure. And the abbreviation for each of those is just to take your number and then put the degree symbol beside it. So this here is discussing the primary structure of the protein. A primary structure of the protein is simply a chain of amino acids. Now, amino acids are our building blocks to our protein. This is our monomer, okay? I've drawn for you an example of an amino acid below. So you have a central carbon, and paired to that carbon, you have an amino group. So this is how it's called, an amino acid. Amino group has nitrogen and 2 hydrogen. You also have a hydrogen attached and a carboxyl group attached, which is a carbon, double bond oxygen, and a hydroxide group. And then you have what's called the R group. Now the R group here is the variable group. There are 20 different R groups. There are 20 different amino acids. And the thing that's different about each of those amino acids is that R group. So here is a picture from the internet of those 20 amino acids that are used in our cells to make our proteins. And if you look, they've highlighted those different R groups, so you can see it's as simple as a hydrogen, or it can be something really complicated like with arginine, okay? Now we're not memorizing this, thank goodness. We just want to make sure that we're seeing, number 1, there are 20 different R groups, so R there is just representing a variation that occurs. And number 2, as we start to look at the tertiary structure of our protein, we will talk about interactions with those R groups. And notice that some of your R groups are nonpolar, so that means no charges. Some of them are polar or charged. And the significance of things that are polar and charged is that if you have charges, then you're going to see interactions between those charges, aka hydrogen bonds. Remember, opposite charges attract and like charges repels, and this will play a role then in our protein structure. So to begin with, that primary structure of a protein is a chain of amino acids connected by a special type of bond called a peptide bond. I like to abbreviate amino acids with 2 little a's, so I've kind of represented my chain of amino acids here. You can see, again, we have another reaction that involves water. So when you're forming the peptide bond, you are doing a dehydration synthesis reaction. And if you are eating a protein and breaking it down into the amino acids, you would hydrolyze or break out those amino acids, those building blocks. Our secondary structure is, let me go back to this. I have an illustration for you. Okay, so we're gonna do this activity in class as well, but this gives you a little preview. I am going to use these beads. They're running away from me. I have just a few beads. I'm gonna use these to represent my primary sequence, primary structure to my protein, which is a chain of amino acids. So just imagine that I've put a whole bunch here. Your sequences of a protein are going to be hundreds of amino acids. And then our secondary level or secondary structure to a protein is described as hydrogen bonding along the length of the chain. And this can result in either an alpha helix, which would be a spring, so coiled up like this. So we can have our protein chain coil into an alpha helix. So you get hydrogen bonding between components of those amino acids. Or sometimes that bonding occurs in what's described as a beta pleated sheet. So that'd be like this, kind of folding, like if you were doing a paper fan, that would be a beta pleated sheet. So something maybe like this. So your secondary structure is folding along the length of the chain, which can be either in the alpha helix form or the beta pleated sheet form. Now the tertiary structure is where we start to see the play between those R groups and the environment. Now our environment of our cell is an aqueous-based solution, and this term aqueous means water-based. Now remember, water is polar, and it's our universal solvent. So whenever we're talking about these proteins being in a water-based solution, that means it's in polar solution, so charge, and universal solvent, the stuff that's dissolved in the water, is all polar or charged. So we're taking a protein which has polar R groups and has charged R groups, and we're sticking it into a solution with charges. And we know that opposite charges attract and like charges repel. So once you take this subunit and you stick it into the cellular structure, it's going to have folding so that those positive charges get as close as possible to other positive charges, and negative charges, I said that backwards, positive charges get as close as possible to negative charges, opposites attract, and like charges get as far away from each other as possible because like charges repel. And so what this means is, depending on the environmental conditions, you can get different types of folding at the tertiary level. So when we talk about the cell controlling that environment of the cell, in part it's being controlled so that the protein folds at the tertiary level correctly. And you would see that each of your subunits would have their own primary chain of amino acids, secondary hydrogen bonding along the length, and tertiary structure. Okay, so we're looking at a protein with two subunits. And then the final level of structure is what's called quaternary. And quaternary I like to describe as protein complexes. So that's when you have different subunits coming together to make your final protein. Now what I've shown here with my pipe cleaners is the globular type. It's called globular because guess what? Looks like a glob. Globular proteins are going to be able to be soluble in water. So when we're looking at proteins that are traveling through the bloodstream, they need to be globular. Your fibrous proteins are going to be those strands. So think of like a hair or a fiber for cloth. Your fibers are the ones that you're looking at in your lab when you see collagen and elastic and reticular fibers. Here's our picture from our text. So here's the whole image of protein structure. So of course we start with our primary structure, our chain of amino acids connected by peptide bonds. So if you've heard the term polypeptide before, that's talking about many peptide bonds. Remember, the primary structure is determined by the DNA sequence. So that's that basic information that's inherited from your parents. Then we see hydrogen bonding will occur between those amino acids. And based on that hydrogen bonding, you're either going to get that alpha helix, which is the spring, or the beta pleated sheet, which is like the folded fan. And then your tertiary structure is that interplay between the R groups in the environment of the cell. So again, when we're homeostatically regulating the cell, that's so that our proteins have the correct final shape so that they work correctly. And then finally, the quaternary structure is that complex, which can either be globular or it can be fibrous. And you know, the key thing here, the significance of this is that in order for that protein to function at its optimal function, it has to have its optimal shape as determined by the DNA and by the environment of the cell. Look here at all the important functions of our protein. So of course we need our enzymes so that we can have our chemical reactions. We need our hormones and our channels and our receptors to be shaped correctly. We need the structure of our body and our contractile proteins to be working. All of these important functions for our protein are dependent on those proteins having the correct shape. Now we're going to be talking about two examples of proteins here in this chapter, and those are the enzymes and the glycoproteins. So we've already mentioned enzymes in a previous discussion. We talked about them as biological catalysts, which remember means they are speeding up the rate of the reaction without being used or changed in any way by that involvement. The other type of protein that we'll mention here specifically is the glycoprotein. And these are the proteins that are on the cell membrane acting as those little name tags. So again, this is how your immune cells recognize which cells belong to you and which cells are foreign or pathogens that need to be attacked. Enzymes, biological catalysts. This means that they are determining a cell's metabolism based on their presence, based on whether they're active or not. That determines what chemical reactions can be happening in the cell. And so the cell will actually control which enzymes are being made, and they'll also control which enzymes are turned on and active. So we'll see that a big part of the communication that's happening with a hormone might be the turn on the production of an enzyme or turn off the production of an enzyme, or maybe it will be to turn that enzyme that's already present on, activate an enzyme or deactivate an enzyme. So we'll see these are all ways that we can change the activity of a cell by changing the enzymes that are available. When we talk about enzymes, we see that they're very specific, meaning that they match up with a particular molecule, which is called a substrate. This enzyme substrate complex is described as a lock and key model. Just like a key is very specific for a lock, the substrate and the enzyme are very specific for each other, and they're binding up based on shape. So the enzyme's active site has to be shaped in such a way that the substrate can bind. So you can think about this as like they're made for each other. When the enzyme pairs up with that specific substrate, then that's how we're going to lower the energy of activation. So here I've drawn my substrate and I have the little line through it. We're going to do a catabolic reaction. So once that substrate binds up, it's going to stress the bond between those molecules and they'll separate into their components, as an example. Your reaction, your enzymatic reactions, again, are dependent on the optimal shape of that protein. So if in any way the shape is not correct, then either the reaction is slowed down a little bit or it doesn't happen. So it's affected by environmental conditions such as temperature and pH, and these have to be at the optimal condition. Now in humans, the optimal temperature would be what we think of as textbook body temperature, right? So 98.6 degrees Fahrenheit. But in the case of pH, this can vary depending on where it is in the body. So if it's in the bloodstream, it's going to be slightly basic. If it's in the stomach, this is going to be very acidic. So it can vary. Now, heat very often will affect protein structure, and it can be permanent. This change of protein structure is called denature, and usually we should think about this as a permanent change. So when you denature a protein, you alter its structure in such a way that it can never go back to its normal shape. So think about cooking food is an act of denaturing the food. All the changes that happen between the raw food and the cooked food, you've denatured those proteins. You've got changes in color and texture and shape, and you can't take a cooked food and turn it back into a raw food.